This is like a rose, they are presenting to it. So this is our naive T cell. They approach these cells as well. But you know what happens? Proposing a girl is not that easy. Okay. So this macrophage basically failed here, but eventually came to this T cell. This T cell was like accepting this macrophage. Okay. Hi everyone. If you are new to this channel, I am Dr. Himadri. Let's talk about the pathogenesis of tuberculosis. In this video, we will be discussing about the pathogenesis part. Okay. So what is TB bacteria? TB bacteria is basically weakly gram-positive and rod-shaped bacteria, and they are non-motile and non-spore forming. And this is the slide you can see. This is basically your Jaden stain or Jehill-Nelson stain. Okay. And this is our bacteria. In the cell membrane of this bacteria, we have mycolic acid, and this mycolic acid is responsible for the acid fastness of this bacteria. And also there is because of this mycolic acid, this is resistant to antibodies as well as complement. Therefore the antibodies and complement cannot destroy it effectively because of this mycolic acid. So this Mycobacterium tuberculosis basically falls under your MTC, Mycobacterium tuberculosis complex. In this complex we have three, there are multiple bacteria, but three are very, very important as they are pathogenic to human being. Okay, the first is your this is your the favorite organism, that is Mycobacterium tuberculosis, and after that we have Mycobacterium africanum, and also we do have Mycobacterium caniti. Okay. So what happened? Look, as per of Indian data, in 2024 there was around 28 lakh patient infected with this TB bacteria. That is, they had tuberculosis, and out of it 3.2 lakh patient died of it. Therefore this is a real danger, right? So the government is basically did what? NTEP, National Tuberculosis Elimination Program, and they invested around $2.5 billion. So therefore, don't even think that the TB bacteria is easy. The pathogenesis is really difficult, but we will try to make it easier, okay, for you. So what happened? Let's see. We are starting the story from here. So we have a baby. So this baby is getting first time exposed with the TB bacteria. How? So look, there was a adult patient of TB. So the patient was coughing. So in the aerosol, you can see this is the diameter of the aerosol, that is 1 to 5 micrometer is the diameter of the aerosol after coughing. And in this aerosol or in this droplet, there was TB bacteria. Usually it contains 2 to 3 TB bacteria. So this is our baby's lungs, who is for the first time getting exposed to it. So these are the TB bacteria entering into the baby's lung. This is the alveoli. Within the alveoli, there was resident macrophages. These resident macrophages are supposed to eat this organism. So how they are actually doing it? Look here. This is our macrophage, beautiful macrophage with open mouth. And this is our TB bacteria. So this TB bacteria actually contains some molecule which are called as CK-like molecule. So this CK-like molecule, and these are your cholesterol molecule present at the mouth of your macrophage. So this rather baby's macrophage. So this interaction made this TB bacteria very, very palatable or tasty. Therefore the macrophage basically eats it. After eating it, this is entering into your phagocyte of macrophages. So this is called your phagosome. But what happens, this bacteria is really naughty. They produce some molecule which are called as TACO. This TACO molecule is basically preventing the fusion between this phagosome and this is your lysosome of macrophages. Therefore, because of this prevention of fusion, this bacteria cannot be degraded very effectively. Okay, so this macrophage is now really frustrated. They try to kill it but cannot kill it. So they travel via these lymphatics and they eventually enter into the lymph node of baby. Okay, so we will expand this lymph node and we will draw it in a big way to know what the actual thing is basically happening here. So look at it. So this is the baby's macrophage, who is for the first time getting exposed to this TB bacteria. So they basically cannot process it effectively. So they try to process this bacteria, cannot process it effectively. Therefore, some unprocessed material like this antigen, these are called as your TB antigen, basically presented with MHC class 2 molecule, major histocompatibility complex 2. Okay, so this is like a rose, they are presenting to it. So this is our naive T cell. Look here, this is another T cell, and look here, this is another T cell. This macrophage, before approaching this girl like T cell, they approach these cells as well. But you know what happens? Proposing a girl is not that easy. Okay, so this macrophage basically failed here, but eventually came to this T cell. This T cell was like accepting this macrophage. Okay, so this antigen presenting cell once proposed with this rose like your red TB antigen. So now this macrophage becomes desperate. So they basically raises one hand. This is your chemical call. It's like a chemical molecule B7. Okay, and there is another hand which is coming from this T cell. Look here, this is CD28. So there is basically the interaction between this B7 molecule and CD28 molecule. Now you can see that the interaction is going very good here. Okay, look here, this is our macrophage. After getting this much of permission, this much of signal, okay, this macrophage is now basically look here, this is interleukin 12 molecule. This is another hand coming from macrophages. Macrophage basically goes one step further with this T cell. So now this bond becomes really tight. Because of this interleukin 12 molecule, what is happening? This T naive cell is basically converted into your or differentiated into your Th1 cell, T helper 1 cell. So this and this flower like molecule is basically CD3. Look here, via CD3 molecule there is stimulation to the nuclear material of this Th1. From this section, what is coming here? Interleukin 2, and from this portion of the DNA, what is coming here is the interleukin 2 receptor. So therefore interleukin 2 is produced by this T helper 1 cell, as well as the interleukin 2 receptor is also produced by this T cell. So they are basically acting in the autocrine fashion. Look here, autocrine fashion. And because of this interleukin 2 and interaction, what is happening? This T helper colony or the specific T cell which is capable of processing the specific TB antigen, basically, so look here, these T cell colonies, these T helper cell colonies basically now produces TNF alpha and interferon gamma. This interferon gamma molecule is very, very important. Basically they mostly produces interferon gamma, but this TNF alpha as well as interferon gamma synergistically act, and they basically acts as like a chemical call. And this is the blood vessel which was passing by this lymph node, by the side of this lymph node, and within the blood vessels the monocytes were running. Okay, so what happens? Because of this, it's like a chemical call to the bro, and these monocytes now receiving this call. Okay, now the monocyte basically jumps from this and eventually enters into the lymph node. And after that, because of this TNF alpha and interferon gamma, what is happening? These macrophages are basically getting activated. Now we call it classically activated macrophages. Okay, so this is also called as epithelioid cell. Why epithelioid cell? Because it looks like epithelial cell, but this is not epithelial cell, therefore we call it epithelioid cell. Okay, so look here, what happened? This is our naughty macrophage, and within the macrophage we have naughtier bacteria. Look here, these are your TB bacteria, and to control the infection, there is what is happening? These are your activated macrophages or epithelioid cells surrounding it, and after that we have what we have? We have this collar of T cells, T lymphocytes, the specific T cells for this antigen. So therefore we call this granuloma, and granuloma is the central thing of, or the central pathogenic part of our. So look here, as we talked earlier that controlling TB is not that easy. So these are our frustrated macrophages. Within the frustrated macrophages we have this TB bacteria, and this TB bacteria actually has multiple pathogenic molecules. So this is your ESAT6 and CFP10 and ammonia. These are soluble molecules which are coming from this TB bacteria, which are basically preventing what they are doing. This is your lysosome and this is the phagosome. But what happens? There are H plus ATPase pumps. This H plus ATPase pumps basically concentrates H plus or the acid within this lysosome. So what happens, this ESAT6CFP10, they basically prevent this or they basically shut down this H plus ATPase pumps. Okay, so therefore lysosome is not effective to kill this bacteria, and we have tachyprotein from our TB pathogen, so they basically prevents the fusion. So the phagosome is not getting fused with lysosome, and another thing is that the H plus pumps are basically getting shut down because of this ESAT6 and CFP10. So therefore killing this pathogen is not very easy within the macrophage. So therefore there is persistence of this TB inside the macrophage, and the TB bacteria will eventually multiply themselves within the macrophage, and the macrophages will eventually getting frustrated. So some part of this TB antigen are literally not very much processed. They via MHC class 2 molecule they expresses. So these are your APCs, that is antigen presenting cells. Okay. So look here what happened in summary. So this is our frustrated macrophage, which we call it APC, antigen presenting cell. They basically, these are like your rows, the TB antigen, they present it to the T naive cell, and this T naive cell, what happens, this APC also produces interleukin 12 as we have already discussed, and with the production of this interleukin 12, T naive cell basically converts or differentiates into T helper 1 cell. Okay, so this T helper 1 cell eventually, after getting activated and differentiated, they produce interferon gamma mainly as well as TNF alpha. This interferon gamma and TNF alpha synergistically stimulates your macrophages and eventually converts it your classically activated macrophage. These classically activated macrophages are also called as your epithelioid cell. Okay, so look here what happens, this T naive cell eventually leads to formation of what? This APC basically produces another molecule, interleukin 1-6 and interleukin 23. This interleukin 23 molecule, these are like the bigger brother. Look, this is 12, but this is the multiplication of 12 by 2 will be 24, but here is, remember this, that this is interleukin 23. Okay, so this is basically the bigger brother which is coming from your APC, converts this T naive cell into T helper 17 cell. So this T helper 17 cell, on the other hand, they produces interleukin 17 and interleukin 22. These interleukin 17 and 22 basically very strong chemoattractant for your neutrophil and macrophages. They will eventually contribute in the formation of the granuloma as well. Okay. What happens? We actually discussed about the granuloma. The granuloma is formed in the primary site. This is your subpleural in location present in the periphery. Okay, subpleural location present in the periphery. This is called as Ghon's foci. Remember Ghon's, the person who basically discovered it for the first time, as well as the frustrated macrophages will travel via these lymphatics and eventually come to the lymph node. There will be formation of granuloma within the lymph node as well. Therefore, this Ghon's foci plus lymphatics plus lymph node is called as Ghon's complex. This Ghon's complex is very, very important. Remember it. Okay. So we need to discuss some points about this TB antigen as well. Look here, there are specific soluble antigen, we have discussed it, like CFP10 and ESAT6, culture filtered protein, and ESAT6 are early secreted antigen target. Okay, as well as this TB bacteria also has some very important thing which is called as LAM, lipoarabinomannan. What happens? This lipoarabinomannan molecule basically stimulates your macrophages, and therefore macrophages produces interleukin 1 and TNF, and this interleukin 1 and TNF are responsible for all those clinical manifestations of this disease like fever, night sweats and weight loss and etc. Okay. So the central pathogenetic part of it was granuloma. Hallmark pathology of this TB infection is granuloma. Okay. So what happens? This TB laden macrophages which is present at the core and which is covered by your epithelioid cell. This epithelioid cell is eventually surrounded by a collar of T lymphocytes. This total thing is actually called as granuloma. So look here, another important thing, this is the normal macrophage. This normal macrophage under interferon gamma and TNF alpha basically converted into your epithelioid cell. which is basically characteristics of our granuloma. Okay, this epithelioid cell or activated macrophage. Okay, so remember another thing, this granuloma is basically caseous necrotic granuloma. What happens? In the center, there is necrotic death of these cells. That is, some macrophages, some bacteria also dies, some epithelioid cell also dies. Therefore, there is necrotic cell death which produces this cheese-like consistent material. Therefore, we call this the caseous necrotic granuloma. Over time, what happens? This caseous necrotic granuloma, eventually there is mineralization, that is calcification inside. We call it calcified granuloma. And look here, another form of this granuloma is, if this granuloma is further more chronic, eventually what happens? There is fibroblast. This fibroblast basically leads to fibrosis, and look here, there is fibrosis, and eventually there is fibrotic granuloma. Okay. So in summary, what is happening here? This is important. Please remember this thing because this is commonly asked in your exam. Okay, what happens? For the first time, TB is a baby is getting exposed to this TB. So if the baby is immunocompetent, which usually happens for more than 90% cases, the baby will develop a state which we call latent TB or persistent TB. Okay. So what is the meaning of latent TB or persistent TB? We just now discussed. That is, the TB bacteria came and eventually the T cells are basically getting primed, and eventually the memory T cells is formed and your Ghon's foci and Ghon's complex is eventually formed. This state where the immune status of the baby is really good, now they basically controls this TB infection. So this is your latent TB or persistent TB, where the immune system basically arrests Mycobacterium tuberculosis at the primary site of infection. So this is characterized by your Ghon's complex. And this Ghon's complex is usually not visible in chest X-ray. Patient is not infectious, patient cannot transmit the microbe, and the patient could be asymptomatic for whole life if the immunity is good. Okay. So remember another state. Suppose a person like a baby is getting exposed to this TB antigen, and this baby, suppose at the age of 1 year the baby is getting exposed, but somehow there is malnutrition around 2 to 3 years of this baby. So because of this malnutrition or maybe HIV, what happens? The immune system is totally down. So this immune system cannot control this TB infection. Therefore, the TB bacteria will overgrow and TB bacteria will reactivate, eventually leading to primary TB disease. This is also called as progressive primary TB disease. Okay. So the criteria for is that for the time frame should be within 5 years of first infection. Okay. To call it primary TB. Suppose something happens like the baby was exposed in around 1 year of age, but at 30 years of age, somehow the baby, now adult, develops HIV infection. Because of this HIV, immune system is once again down, or maybe the person is receiving some chemotherapy because of some malignancy. So what happens? We call this state as post-primary disease, because the criteria is it should be more than 5 years of first infection to call it post-primary disease. So now discuss about the complication of this pulmonary TB. What happens? So this is our lymph node which eventually getting infected because of this TB. There is enlargement of this lymph node. So this it is pushing this airway. Okay, compressing this airway. During inspiration, air can easily enter in, but during expiration, the airway becomes narrower. Normally it is getting narrower, as well as it is getting pushed, and therefore what happens? There is air trapping. Okay, during expiration all the air cannot come out, therefore there is air trapping. This is the first complication. And secondly, what can happen? Look here, suppose the airway is getting compressed too much that it is getting no air can enter during inspiration, and also during expiration no air can go out. Therefore we call it the condition which is called as collapse. So number one complication could be air trapping, number two collapse if it further goes and further compresses. Okay, and number three, what could happen is that, look here, the necrotic material can come out into the bronchial tree. Therefore, we call it endobronchial TB. Okay. And what can happen? This necrotic material with the bacteria can eventually enter into your pleura and eventually leads to pleural effusion. Okay. And suppose another thing, the fifth complication is that suppose this is our pathogenic organism and this is the primary Gons foci. So what happens if the immune system is down? The primary foci will increase in size. There will be more caseation, and caseating material eventually will be converted into liquefactive material. There will be clinical features on all the clinical features of TB. We call this stage as progressive primary TB. Okay. So, and another thing which can happen, look here, via lymphatics, from this lymph node, the TB bacteria is eventually coming, and eventually they enter into the blood circulation. If the TB bacteria load is too much and your immune system is down, TB bacteria from the lymph node is eventually coming into the blood circulation and proliferating within the blood circulation. We call this disease as disseminated TB disease. Disseminated means the TB bacteria within the hematogenous route. Okay, so there will be high grade fever, the baby will be sick, there will be the lymph node will be enlarged and all that, there will be hepatosplenomegaly. And suppose this organism basically infects two or more than two organ system via hematogenous route, we call it miliary TB. The classical presentation, the classical thing which we can find in the chest X-ray is the millet-shaped granules in bilateral lung field, which are usually less than 2 to 3 millimeter in size. Okay, so this is important, and these are the organs where TB actually lodges, rather loves to lodge, in the brain, in the kidney, and in the bone. If you are still watching me, you could have liked this video, and for further and more videos, more interesting videos later, consider subscribing and consider sharing with your friends. Thank you.